In the video. Should we have the video ready just like right now? Just like no one is. Yeah. You think so? Would it be nice to have the video already right now? But not like.
All right, thank you all very much for coming. I'm not sure if you can hear me with the microphone, but thank you all very much for coming to CQ-AState. Thank you. We're now going to hear some opening words from our very own Dr. Juarez.
Thank you. I'm going to use the microphone for the lovely people on YouTube because I think my voice projects rather well from here, right? Yes. Oh, there we go. I am tiny, so. Well, thank you everyone for coming today, but especially thank you to all the students that are going to present their hard done work, their projects. Most of you are seniors. As you know, CQ-AState is a tradition in Arkansas State. There is an equivalent CQ-AState taking place in Jonesboro at this very moment. And our young CQ-AState at Campus Querétaro has been a tradition where the students that are graduating present the fruits of their work, right? This year, for the very first time, we are having only research of original products, original work, which is, I think, a step in the right direction for our CQ-AState and for our young campus. So you should all feel very, very proud of the work that you're going to be sharing with us today, tomorrow, and on Friday. So props to you. And a special thanks to Dr. Katy, who has been organizing for the third time this year. And for the first time, Dr. Bernardo Pérez Zamorano is joining. And I am sincerely hoping, wink, wink, that this is his first, but not his last. And he continues with Dr. Katy's trend of 3 out of 3. So thank you everybody for participating this year at CQ-AState. You are the people that build the event. So this is a great opportunity to share what you've done. Be proud of the work that you are presenting. This time around, you get to do it at home, right? Next time around, probably in a bigger place. So it is special to have you here to show what you're doing. And I want to give a big shout out to our sponsors in Copper. They have very generously prepared something for each of the students that are going to be presenting. And especially, I want to thank Julio because I am especially proud to see him as a judge today because he was a student a few years ago. So it's lovely to see you come full circle. And without further ado, let's inaugurate. Let's start with our amazing CQ-AState 2024, and let's enjoy some knowledge.
Before we begin, we'd like to all extend a shout out to Coppert for being so gracious as to fund this event and for funding the gift bags that will be distributed throughout the event. Thank you.
Most farming challenges can be found in Asia. And we've been developing these solutions for over 55 years. Our biological products help farmers to protect crops from pests and diseases while encouraging more efficient growth. The answers are already there, so why not use them? Corporates, partners with nature.
Okay, and our very first presentation will be symplectomy, I apologize, effects on intra-abdominal organ catecholamiine levels in streptozotocin induced diabetic rat model, done by Andrea Giovanna Aguirre.
To go next, up to-
So, next.
Down is next, and there's a laser pointer.
So, hello everyone. Good afternoon. My name is Andrea, and I'm glad to be here to talk about sympathectomy effects on intra-abdominal organ catecholamine levels in a streptozotocin-induced diabetic rat model. This work has been published in Life in 2022. So, diabetes is a major public health concern, and it is basically a disorder in the metabolism of carbohydrates, lipids, and proteins that is associated with relative or absolute insufficiency on insulin secretion. And well, one of its most common complications is autonomic neuropathy. And this arises when there is damage on the nerves that control the automatic body functions. And well, this can also lead to sensory, motor, and sympathetic dysfunction in approximately 51.9% of patients. And now we are going to talk about the sympathetic nervous system.
O sea, ¿cómo afecta? Me imagino que tiene que ver lo de química, ¿cómo afecta todo el sistema?
Es un proyecto con mi hermana.
Ah, ya.
Hice una publicada en un artículo. Y pues ya tiene. Sí, yo creo que se salió, ¿no?
No sé, se debe haber quedado así como pasmado. Que luego se parte la comunicación, ¿no?
Ujú.
Yeah. So I was saying, now we are going to talk about the sympathetic nervous system. It is part of the autonomic nervous system, which is in charge of controlling the automatic body functions like the blood pressure and temperature. And the sympathetic nervous system is best known for its role in responding to dangerous or stressful situations. And this response is precisely mediated by the release of catecholamines. And well, catecholamines are hormones that are secreted by the adrenal glands, and they also play important roles in the regulation of various physiological processes. And well, several studies have reported alterations in the concentration of catecholamines, particularly those of epinephrine, norepinephrine, and dopamine. And this has been directly linked to complications of diabetes. Now, you may be wondering, how can this be assessed? Well, most experimental settings use drugs that allows us to study the relationship between the sympathetic nervous system and diabetes. One of those is guanethidine. Guanethidine is a drug that induces a blockade in the sympathetic nervous system and hence produces alterations in the release and the storage of catecholamines. And well, diabetes is also commonly induced by using streptozotocin. And streptozotocin is a drug that is toxic to beta pancreatic cells. And well, these beta pancreatic cells are essential because they are in charge of the storage, the production, and the release of insulin. It is not, can you help me, please? So the objective of this study was to analyze the interaction between diabetes and the sympathetic nervous system by building a model of sympathectomized diabetic rats to assess alterations in the concentrations of catecholamines in different intra-abdominal organs. Now, before going through the methodology, I need to tell you that all procedures that are described in here were set up according to the the Ethics and Biosafety Committee of the National School of Biological Sciences in the National Polytechnic Institute, as well as in accordance to the official Mexican guidelines for laboratory animal use and care. So the first step was the animal preparation. We had 24 male Wistar rats, and they were randomly placed into 4 different groups of 6 rats each one. And well, of course, the control group received no treatment. One group received a dose of guanethidine, which was the sympathectomized group. Another group received the same dose of guanethidine plus 1 dose of streptozotocin to induce diabetes. And finally, the diabetic group only received a dose of streptozotocin. So in order to assess the different biochemical parameters, blood samples were taken from the rats. They were centrifuged so that the serum could be separated, and the serum was analyzed for glucose, cholesterol, triacylglycerol, and albumin. And this was done using diagnostic kits, just following the protocols. And now for the catecholamine levels assessment, different samples were taken from the tissues, and they were analyzed using high performance liquid chromatography that was coupled to electrochemical analysis. And finally, the effect of the treatment was analyzed using a statistical software that was, in this case, the SPSS software. So now we are going to see the results. This table shows the different biochemical parameters and the different groups. As you can see, we have glucose, triacylglycerol, cholesterol, and albumin. And the asterisk indicates significant differences with respect to the control group. So as we can see here, for the case of the diabetic groups, is to say the ones that were treated with streptozotocin, we can see that there is a significant increase in the levels of glucose, triacylglycerol, and cholesterol with respect to the control. And it is enormous. For example, here in the diabetic group, it was 508 compared to 98 in the control group. And in contrast, albumin levels show a decrease, of course, in the diabetic groups compared to the control group. Now we are going to see the figures representing the different concentrations of catecholamines. For the case of norepinephrine, we can, also here, the asterisk indicates significant differences with respect to the control group. So in this case, we can clearly see that the lowest concentrations of norepinephrine were found in the liver of all groups. And well, the other relatively low concentrations were found in the pancreas and in the heart of the groups that were treated with streptozotocin, it is to say the diabetic group, and the sympathectomized and diabetic group. Now, regarding epinephrine, here we can see that the lowest concentration was found in the heart of the group that was treated with guanethidine, or the one that was sympathectomized. And another low concentration was found in the liver of the group that received guanethidine and streptozotocin. And also, low concentrations were found in the heart of the group that received the same treatment with guanethidine and streptozotocin, or precisely the group that was both sympathectomized and diabetic. And now regarding dopamine, overall we can see that the lowest concentrations were found in the liver, the pancreas, and the heart of the different groups, whereas the highest concentrations of dopamine were found in the paraganglia of the different groups, and also significantly higher concentrations were found in the portal vein of all groups. And finally, this figure shows the concentrations of the different catecholamines in the adrenal medulla. As can be seen, the one that showed the highest concentration was epinephrine in all groups, followed by norepinephrine, and the one that showed the lowest concentration was dopamine, and well, the group that showed the lowest concentrations out of them all was the one that was sympathectomized, or the one that was treated with guanethidine. Now, regarding the discussion, well, as you could see in the results, the elimination of the sympathetic nervous system, which basically took place on those groups that were treated with guanethidine, showed a significant increase in the levels of glucose, triacylglycerol, and cholesterol, whereas there was a significant decrease in the albumin levels with respect to the control. And also, out of all the catecholamines that were assessed, well, norepinephrine showed the lowest concentrations in the liver, as it was clearly seen in the figures. And the content of epinephrine that was found in the heart, in the liver, and in the pancreas was consistent with the data that is reported in the literature, because epinephrine is only found in peripheral tissues. And lastly, the fact that the dopamine concentrations were not that consistent among the different groups may be explained by its rapid transformation into norepinephrine, because dopamine is the precursor of this catecholamine, norepinephrine. So summing up, the results indicate that the effect of quinetidine, which is the one that causes the sympathectomy, as well as the effect of streptozotocin that leads to the induction of diabetes, were significant in the final levels of blood glucose, triacylglycerol, and cholesterol. And likewise, well, the different alterations in the catecholamine concentrations that were observed may be easily attributed to the duration and the severity of diabetes. And well, last but not least, I would like to thank the National School of Biological Sciences of the National Polytechnic Institute in Mexico City, as well as Dr. Angelica Perez Juarez, who is also a co-author of this study, for her guidance in all the experimental setup. And well, thank you.
For our second presentation, we'd like to invite, oh, that's right, my bad. Do we have any questions?
Yes. Thank you.
A very high performance. Again, to the main researcher, it's impressive. Yeah, I would like to ask you two things. First one, you mentioned that even only many times.
Yes. Male wizard rats are like the standard, like the ones that are commonly used. And we use males mainly because they have like less hormonal fluctuations, and they allow you to work better with the trials in the experiments.
So you would say that male rats are like more metabolically stable? That's
Yes.
kind of the idea?
Like they are more suitable.
Ah, they are more suitable.
Yeah.
And it's because of the metabolism, or it's because of I don't know, because I am not so aware of why you're using it. And if you repeat this experiment, would you still use male rats, or would you use like a mix, or only female?
No, I would say that, well, since as I mentioned, since using male wizard rats is like the standard, if I have to repeat it, I will like follow basically the same guidelines, the same protocol.
Okay. And my second question is actually more personal, if may I. What would you like to pursue after graduation? Because I think you would be an amazing researcher.
Yes, thank you. Yeah, maybe a master's, but like my preferences are more oriented into food science or the biochemistry part.
Very good. Thank you very much.
No, thank you.
Hello, Andrea.
Hi.
Fantastic, really interesting work. I wanted to ask you something more about not just the research and so on, but more about the experience that you had. How long did it take you to make all this research? And that is the second question. How did you felt in working with rats? And what was something completely different, isn't it?
Yes. Well, first of all, answering your second question, I was not like in direct contact with the rat, because my highest contribution was with the processing of the samples. So I was not like in direct manipulation with the specimens. But regarding the time that it took, if I'm not mistaken, it took us about, well, the treatment was given for 6 weeks, but like all the experiment took us like 8 weeks, almost 10 weeks.
So what's a kind of summer
Yes.
over there?
Yes.
Fantastic. And I guess that then all the analysis and statistical analysis was what took ages.
Yes,
time
yes,
and
yes.
everything.
And also like sending the draft because as I mentioned, this one is published. So like the part of the drafts and the writing, yeah, it's a whole thing, yeah.
Fantastic. Congratulations.
Thank
because
you.
You're finishing your undergraduate with a paper. So
Yes.
it's an amazing work that you've done.
Thank you. Thank you.
Thank you very much.
And Dr. Satarain had a question.
We have one question online. Can we? Yes. Please, Marco Alvarez, if you want to.
Yeah, thank you so much for your presentation. After your study, what is the most important result that your team found in the association of diabetes and sympathetic neuron inhibition? For continue with this important discovery that you show, because the paper is now published, so I want to ask you, what is the next step?
Oh, maybe first of all, thank you, Marco. I guess the next step would be to repeat the whole process to ensure to check that the results are consistent, and then we can apply other type of essays to continue in this line of analyzing the association of diabetes and the different catecholamine levels. So I would say that that will be the next step.
Okay, thank you.
Thank you.
Andy, congratulations. It's
Thank you.
really amazing work. And I know that working with animal models is tremendously difficult on many levels. So I want you to tell us a little bit more about the actual experimental model and the way that you controlled the data acquisition, particularly how you actually acquired your tissues and the type of treatment that you gave them in order to be able to measure out using these methodologies that you have mentioned previously. And also considering all of these controls that you took, what do you think then is the actual source for seeing these large differences between the different organs that you're looking at? Is it that the hormones sort of get drawn into that area, or is it that these tissues accumulate more blood and then by the time that you took them out, they were more filled with blood as opposed to adjacent tissues? Or what can you conclude from your model and its limitations and strengths?
I would say that the results came mostly for the accumulation of blood and the accumulation of catecholamines in the different organs, but also this can be an area of opportunity, like in future replications of the experiment. And well, regarding you asked about the treatment of
how you acquire the tissues, like the process, what type of methods you use for the euthanization of the animals, how then they were treated in order to acquire the tissues, where did you place the tissues, how did you process your samples in order to get the data?
Oh, basically, well, it is a really detailed protocol. So it is basically everything is explained in the paper, but summing up as a summary, for example, for the part of the catecholamine levels, the tissues, some samples were taken from the tissues and they were, if I'm not mistaken, they were treated with perchloric acid and they were placed at -70 degrees until they were going to be used. And then we use the HPLC, which is, and we basically charge the samples there. And we used electrochemical and also spectrophotometric techniques to assess this part. Yeah.
Thank you.
No, thank you.
Well, thank you for your presentation, and it was quite amazing the work you have done. Actually, looking into the norm 062 ZU 1999, it actually specifies that you can use several animal models, such as, like you say, rats, but they also say that you can use carnivores or porcines or even primates. Do you think that you can actually use another animal model for this experiment?
Yes, it may be possible. I mean, in this case, we use rats because, as I mentioned, I worked with my mentor, and the fact she has used rats basically since she was in the masters. So it's like an standard in her work. So basically, we followed that guidelines or that protocols. But yeah, it would be interesting to use another different animals like carnivores, etc.
Okay, now actually, we would now like to invite Natalia Reyes Vasquez to the stage for a presentation on the synthesis of analogs of CSC 02748044 to work as inhibitors of DNA methyltransferase DNMT1.
Hello, everyone. Welcome to this to my final presentation, which talks about the development of a synthesis of a library of compounds to work as inhibitors of DNA methyltransferase 1 or DNMT1. Well, first about what is the process of a basic summary of what is the process of DNA methylation. Basically, this is an epigenetic process in which it works a lot for gene regulation. It basically turns off around certain genes. But how does this process happen specifically? Well, with these enzymes called DNA methyltransferases, in which they use as a cofactor S-adenosine methionine to transfer the methyl group. In this case, we have cytosine to the 5th carbon of the cytosine nucleotide, and leaves as a byproduct is adenosine homocysteine. We are going to talk a little bit about it later, so let's keep an eye on this one. And well, basically, there are two processes that can happen in our bodies, which is hypermethylation, which is sites that usually are not methylated, get methylated, and therefore the gene turns off. And also it happens a process called hypomethylation, in which sites that are usually methylated, called cytosine and CpG sites, cytosine and guanine sites, get demethylated, and therefore these two processes contribute to the development of cell differentiation or even tumorigenesis. So there comes our interest in these enzymes and how to inhibit them. So right now, there are more than 256 compounds that are in chemical databases that have measured activity against this enzyme, but only 2 are FDA approved, which are azacitidine and decitabine. The problem with these 2 compounds is that they have low specificity, they have toxicity, poor bioavailability, and even instability in physical conditions. So this project actually started in UNAM with a group called DIFAKIM, in which their main goal, was to identify DNMT inhibitors with novel chemical scaffolds, which means that they have a different or new structure that was not previously recognized as an inhibitor, in order to enhance or to prove their inhibition against the enzyme. They use this enzyme, which is the crystal structure of human DNMT1, and here we can see the active site. So they used 10 compounds, and they did all the molecular docking and even some biochemical assays, and they found 10 compounds that have novel were good as inhibitors of this enzyme, and 4 that have actually novel chemical scaffolds not registered before as possible inhibitors of the enzyme. So from here, we chose one, which is this compound, SCS 27480404. It has this structure. We can see that it has a good percentage of inhibition, 30%. The molecular docking score was minus 9.3, and the CIF is 5.84. From here, we also have a comparison between the chemical space against its natural. This one is the natural inhibitor of this enzyme, is adenosine homocysteine of the DNA methyltransferase 1. So the part here that is ASU-CQ starts with how to synthesize this compound. We used a disconnection approach in order to develop an initial synthesis, but it wasn't working that well. And then this project got traveled all the way to Barcelona, in which they they developed a new synthesis. The goals of this project are to determine the most convenient way to synthesize our target molecule or analogs with the same or improved inhibition against the enzyme, and also develop a synthetic method to synthesize a library of compounds. So here we can see the optimized synthetic pathway in Barcelona by Miss Diana Prado. Here, by Miss Diana Prado. Here we have first a nucleophilic addition with that ends in our first building block, which is this one. Then we have an esterification of this compound that finally gets coupled with palladium to the protected piperazine. Finally, the piperazine gets deprotected, and we do a final coupling with these two in order to obtain our target product or our analog. So from here in Barcelona, 3 compounds were synthesized, DLP 13, 18, and 19. They have yields from around 20 to 50%, and DRFs are between 0.4 and 0.3. DRFs is how much the spot runs in the TLC. This one is gonna be important for us because we're gonna compare with our results done here at the SUCQ. And here we have a characterization of the DLP 13. So sorry, but we here is the proton NMR of this compound DLP 13. Some important peaks to highlight is this one, which is the one of the protonated nitrogen. We also have this one at 1.4, which is the one of the 3, the final carbon here of the ester. At 4.4, we have the other one here of the second ester. And finally, here at 3.2, we have a singlet that is actually this carbon over here. And we also got the exact mass. This is a process that helps us to see if we actually have the desired mass. Here, as we can see, the exact mass is 385.18, and it cannot be seen really well. But here, we have a mass of 386.18, which is actually taken into consideration that we added a hydrogen. So we have a really good result regarding this assay as well. Finally, we also did a biological evaluation against the enzyme. Well, they did in Barcelona. And we used as a positive control the SAH. And here we have that DLP13 actually did not show inhibition, but the other two showed a maintained inhibition against the enzyme. Finally, the work traveled all the way back to here at the SUCQ, in which we were able to synthesize 6 products that are going to be tested to be possible inhibitors against the enzyme. In the first one, in this table, we can see that we modify both parts here, the blue part and the yellow one, of the target molecule. We used thiazoles and isoxazoles and even benzenes instead of this original part. And we also use benzene in this part instead of naphthalene and an ester. We can see the yields are between 20 and 5%, and the RFs are between 0.4 and 0.3, really similar to the ones synthesized in Barcelona. Also, we finished another 2 compounds, which have the NMT 15 and 14, now keeping the naphthalene instead of just having the benzene, and also having the same differences in this part. And in here, we also have the esters. And we have a yield of 27% and 23% respectively. And we have an RF of 0.65 and 0.5. So from all of this information, we can make some conclusions. First, that we have an optimized synthetic pathway, and that this is proved by the characterization of the compound, that actually the synthesis works. We also have a biological characterization that shows that the compounds maintain the inhibition. And finally, that we have started a small library of compounds. So for future work, all of our compounds were now sent to the UNAM and are being analyzed for NMR, possibly exact mass, and even biological tests. Also, we want to test the biological activity of these other compounds that could possibly work as inhibitors, and do the molecular docking evaluation as well. And finally, to continue to play with the structure activity relationships of the compound that we talked about today and this other compound that shows also a good percentage of inhibition and promising also docking score and promising a SIF. And well, finally, I want to thank Dr. Federica Cati, my supervisor, also Dr. José Luis Medina Prado, he is the leader of the DIFAKIM group at UNAM, also Ms. Diana Prado, also Ana Karen Alexandra Campos de Luna, she worked with us for the Verano de la Ciencia project, and finally all of my coworkers, Edna, Claudio Luz, Daniel, and Mariano. And also the University of Barcelona and the DIFAKIM group at UNAM. And well, that's it. Thank you very much for your attention. And if you have any questions, I'm happy to answer.
We'd now like to open the floor for questions.
Natalia, thank you for your presentation. That was quite interesting.
Thank you.
I'm wondering if you could tell us a little bit about what you feel the overall purpose would be to synthesizing and being able to carry out these biological tests, what additional tests you would need in order to go to the further step, and what you're thinking about in terms of how these would be used for pharmacological purposes in humans.
Thank you. Well, first, the part of synthesizing not only the target molecule to synthesize other ones is that we talked previously with this group at UNAM, and we proposed some other structures like the ones that I showed today that we synthesized, and they did their own molecular docking, and they told us that these structures could work with an enhanced effect or maintain the inhibition effect. That's why we thought that it was a really good opportunity to synthesize them and to see the biological effects now experimentally. And I think that for future work, we also have other structures that need to be also enhanced and even find a synthesis for them, because right now we don't have it. And well, I think that the main purpose is to, at some point, if we find one that is good enough, maybe even go to a clinical trial. But as we know, developing a drug takes a lot of time. So that is like a more in the future part of the project.
Thank you.
Thank you.
Thank you, Natalia. Congratulations, amazing work. I mean, I am impressed with the amount of work that you and Dr. Cathy and your peers actually perform in the lab. I know that synthetic pathways are complicated by my own experience, so congratulations.
Thank you.
Could you go please show us the synthetic pathway, the optimized one, please?
Yes. It's not moving.
Thank you very much. So the protection with the bot, where is it? Where is it?
Yeah.
At
Here.
the bottom, thank you. So usually, I mean, BOC is fantastic to protect amines, right?
Yeah.
But sometimes, you can compromise the nucleophilic part of another side of the molecule.
Yeah.
Because by esterifications, as you know, you can hard the electrical density of the molecule, compromising then the nucleophilic power, and thus diminishing the chemical potential. Okay. So, did you notice any setbacks over that nucleophilic attack at that step when adding 5 to 6?
No, actually, well, experimentally, we did
Yes,
have
experimentally.
a problem with this part.
Uh-huh.
Because actually at the in our first synthesis, this one was not protected, and we showed that then the final coupling didn't happen, probably because this process happened twice. So then this coupling didn't happen because these two nitrogens were already full, or they were already used. So that's why the bulk group helped us a lot to only do the attack in one of the nitrogens, and then after ensuring this bond, then deprotecting it to do the final coupling part.
I see. Very good. Very good. And it's a lot of steps. Which was the most challenging to purify?
The last one.
The last
one.
The
is
target
really
molecule was
hard.
the what? Okay.
Yeah, we actually purified them by column chromatography, and it took a while. Yeah. Two compounds we tried and we synthesized them, I think, twice, and we tried twice, and then I just didn't want to purify.
And why do you think those were difficult to purify or to obtain? Because I'm sure you had problems with the column, right?
Yeah, it is still in the column.
They say like,
Yeah.
which
Are the ones that are not shown in here, I think, is the. Sorry. These two, this one and this one, we tried, but we didn't work.
have you think what did you use alumina or silica?
Silica.
Have you think about maybe using alumina?
No, we haven't, but it could be a good idea to
Maybe,
try.
because since alumina is basic, and I think that the problem is that the both three nitrogen, you have three nitrogens, right?
Yeah.
So maybe those three nitrogens are the ones that are attaching to the acidic part of the silica. Okay, so maybe using one that is basic like alumina
Yeah.
might help you in obtaining better purifications. We do have alumina here in the university, and now we have the foreigner, mofla, in English, the oven, the oven.
Yeah, the oven.
We have the powerful oven, sorry, in which we can activate it. Okay, so maybe it's a possibility that you can explore. Okay,
Yes,
and
thank you.
finally, could you please go to the mass acquisition, please?
Yes. This one?
Yes, the spectrometry. This tells me, first of all, wonderfully obtained. I'm impressed.
Yes, thank you.
This is impressive. You have mostly one signal. And this also tells you a lot about the stability of your molecule. Okay.
Mhm.
Which technique was used? Do you know?
Yes,
Ionic
it was
impact?
ionic, ionic electrospray, I think.
Okay.
Yeah, it was.
Okay. It's impressive that a molecule that it's so small, it's actually so stable under so hardish conditions.
Yeah.
So that also tells you a lot about the stability of the molecule, which also tells you a lot about the drugability of the
Yes.
molecule. Okay. So congratulations to you and to Dr. Pet.
Thank you. Thank you very much.
Well, amazing presentation by you. I just have a quick
you.
question. You might know that you cannot patent molecules, but have you explored to obtain the pattern of the method that you actually are synthesizing? Because I mean, if you're basing your results, you may have an original synthesized pathway in order to apply to a patent to these molecules.
I think that's definitely a step that we have to do in the future, like patent the synthesis, because it took us a while, and also it was not only done by us; it was also done by Ms. Diana Prado at UNAM. So we also would have to talk to her about it and to see how can we develop this patent together, maybe, or with her.
Thank you very much.
Thank you.
If there is anybody in the Zoom that want to ask anything? Otherwise, we can close.
Well, thank you very much for your attention.
Don't worry, don't worry. It's okay. Don't worry. On the show.
Excuse me, did you have the other microphone? All right, for our second presentation, we will have Rebecca Anguinao, Susana Coronel, Hugo Castañeda, Luz González, Andrés Oramas, and Grecia Palafox, giving a presentation on exploring tenebrial Molitor's antifreeze protein, evolutionary insights, and cryoprotective applications, part 1 and 2. Okay, so hello everyone. We are Rebecca, Susana, and Hugo, and I don't know why I can't. Sorry. No. I don't know. Okay. So we will talk about exploring Tenebrio Molitor's AFP evolutionary history. AFP stands for antifreeze purring. So first of all, we have to define what is Tenebrio Molitor, which is here. Tenebrio Molitor is a yellow mealworm beetle that is characterized for being freeze susceptible. This means that it needs the antifreeze proteins for survive at low temperature. So the thing here is what these antifreeze proteins do. Basically, these
Pues me saque y entra, no sé por qué.
¿Sabes qué tienes? Prende tu micrófono.
don't worry. So, as I was saying, the Tenebrio molitor is a yellow mealworm beetle that is characterized for being freeze susceptible. And basically, it needs from the antifreeze proteins for survive at low temperatures because the antifreeze proteins, what they do is that protect the cells of different organisms, such as the Tenebrio molitor, by avoiding the binding of the crystals, ice crystals, from these low temperatures. So basically, we have here the structure of the Tenebrio molitor antifreeze protein, which consists in 85 amino acid residues, and we can see it's a very small protein. And it is characterized for being beta helix right-handed structure. And in each helix that it is important to mention that has 7 loops, contain 12 amino acid residues. The most common amino acid residues that we can find are cysteine, which will provide like support to the structure by making disulfide bonds, and threonine, which will provide the ice binding surface for the ice crystals to binder. And following, we will talk about the potential uses of Tenebrio molitor AFP later.
Okay, now talking a little bit about the methodology. First of all, I will explain this part to just give a panorama, So first of all, we use BLAST, which is basic local alignment search tool. Here we look for our alignments, homologous alignments. Then we use the alignment for this ones. We use Cluster Omega and MAFT. And then we did our IQ tree to find the best model. The PyML or phylogenetic maximum likelihood for the preliminary tree. Then the 3-time ancestral. We use AlphaFold we use this to obtain the 3D structures of the ancestors. Mystic or mutual information survey to infer coevolution. Here we use for find the coevolution of the ancestor proteins. And finally, we use Benchling, which we use it for biochemical properties, to find the biochemical properties. Now, here in the first table, we can find the BLAST alignment. Here in the description, we can see the species. Here we can find the query coverage and percentage identity. The important thing is that the ones that are on the top are more identical to our query, which is like our seed for our alignment. Here in the figure 3, we can find the closal omega alignment. Here in the gray part, we can see that they are identical. In the red part means that they are not identical, but similar in structure. And then in the blue lines, they are completely different. and the red lines here means the gaps that we find in our sequences. In the table 2, here we can find the IQ tree. That our best result was flu G4. Now, in our figure 4, here we can find the phylogenetic tree that we obtained through 3-time ancestral. This is the tree of AFPs. Here, it's important to mention the species of the beetles, as all are beetles that make sense, which are good results. Here, these dots represent the relationship with this, the nodes of these protein structures here, the 3D protein structure that we use AlphaFold for this. So, and we of course use color codes for be more easy to relate them. In the structure, we can see here a variability in its ellipses. And if you remember in the structure that we are using of AFPs, it's way shorter.
So all the sequence were submitted to Mystic, the ancestors, and also the sequence that we search through BLAST in order to obtain the coevolutionary through a mutual information statistic analysis. That is represented in this circus diagram that is basically a multiple sequence alignment that we already have seen, where the blue color stands for a less conserved amino acid, and the red color stands for a more conserved amino acid. Here we also can observe the amino acid and the position of the amino acid in the whole protein. Then, the internal lines. As I already mentioned, this Mystic, it's in order to understand and observe the coevolutionary relationship between each amino acid with other amino acids. That means that basically whenever one of these amino acids change, it has a tendency, it has been in the analysis showed that it's a tendency of the other amino acid in which this is connected to also change. It doesn't mean that it will change, but it has that tendency. Where the red lines that we can observe from 59 to 41, and also the couple of 25 to 47 are the top 5 highest scores. So that means that for almost all the sequence, it was found this relationship. And then the black wider lines are the 70 to 95 score. And we have the remaining with that line that you can barely see. Okay, for the bioinformatics, I mean, we studied the biochemical properties, but all theoretical, as they were only ancestors that we calculated using the IQ tree on the process we already mentioned. So we obtained that the molecular weights were much higher, much larger than the original AFP, that it's only about 11.9 kilodaltons, while the isoelectric point, that's it's basically when the protein has a neutral charge, the protein has a neutral charge under those pH conditions, where we can observe ancestor 2 and 3 to have more similar to blood, and also the instability index. We have to understand from here that when this value is less than 40, it means that it is stable. So most of the molecules that we obtain, even the original AFP, are very unstable, so they can denaturate easily and lose the shape. And finally, for the hydrophobicity profile, using the Kaido-Little hydropathic profile, we obtain the largest values that are around 3.5, but that doesn't mean nothing if we don't take in consideration that actually is the surface of the protein, the one that is in contact with water, right? So what we can see here is that the blue patches in the surface are more hydrophobic, and the white are neutral, and finally the yellow ones are hydrophilic. So in overall, most of the protein is hydrophobic. So what can we conclude from these results? First, that some of the ancestors of the AFP have a greater thermal stability, so they can be used for other purpose, right? Also, taking in consideration the isoelectric point that for medical research, for medical purpose, they have to be closest to the pH of the blood to maintain that net charge of 0 with the isoelectric point. And some of the answers, so as we already mentioned, have this pH of 7.4. The residue pairs, when we see the circle, we see how they were related, these pairs, 25, 47, and 41, 59. So that means that they are very important to the maintenance of the structure of the protein, or even for the function of the protein. So they are prime targets for protein engineering, so we don't mess up with all the protein. And finally, as I already mentioned, overall, the protein seems to be hydrophobic in the surface, so they might be able to bypass the cell membrane. Also, we have to take in consideration the very low molecular weight.
So now, for future work, we propose modifying the current tenebrum molecular AFP to enhance its temperature resistance using the ancestor sequence in comparison.
And we also want to assess that it is important to take in consideration that TM-AFPs could be used in different industries and in different fields, such as the biomedical, food, and environmental industries. And also, it is important to know that there will be future research for modifications and for assess the impact on the protein functionality and stability. Thank you. The floor is now open for questions. Okay.
So hello, I'm Andres, this is Grecia, this is Luz. Today we're going to be presenting tenebrum molitor's antifreezes proteins cryoprotective applications.
Okay, so tenebrum molitor's antifreeze protein is one of the most multidisciplinary proteins in the world. This means that it goes from food industry, space engineering, agriculture, and finally, and the most important one, medical industry. In this field is one of the most important ones because AFPs can be used as cryopreservation, And one of our main objectives for this research was to determine the viability of tenebrum molitor antifreeze protein in cryoprotectant role. Okay, so now I'm gonna explain a little bit our research. In this case, we decided to divide our research in 3 different parts. First of all, in expression test, survival test, and So first of all, in our expression test, it is important to mention that our gene was already inserted in a vector called PICL1. This vector, it has the characteristic that is only activated at low temperatures. Also, we decided to use the VL21 E. coli strain because it is the most useful one for protein expression. And also, we decided to do our experiments in several acrylamide gel electrophoresis.
Okay, so for our first trial, we decided to use BL21 cells without the plasmid as controls. We also used, well, we had tests for warm and cold, to analyze them. And we found out that, well, we also used BL21s, but in this case, we used the plasmid and it was incubated, the warm ones at 37 degrees Celsius and the cold ones at 4 degrees Celsius. What can be seen here in these both tests was that we did not find a very noticeable or a very important difference between our controls, warm and cold tests. And we, however, managed to express a lot of proteins And although this was not a lot of information regarding the temperature or how it was affected by it, we did manage to find out how much of the protein we needed to put in the samples in order to get like good bands instead of everything smeared.
So following, as you can see, we decided to change our method and we decided to implement IPTG, which is a lactose analog. This is because our first vector, pickled one, it had a lactose operand. So also we decided to implement to use DH5 alpha E. coli strains, and this is because we read in the literature that DH5 alphas were used for AFPs. Also, we decided to use several survival tests with 3 different conditions for 1 hour, which were -80, -20, and -12 degrees Celsius. And also, we decided to implement lysozyme to start lysing our cells to do the acrylamide gels.
Okay, so using IPTG, in this case, we did manage to express protein, as it can be seen in all of these tests, and we did manage to get a band around the size that we wanted, which was 12 kilodaltons. However, what was troubling about these results is that gels A and B were with samples that were placed in cold temperatures, and C and D were placed in warmer temperatures. So as you can see, we do have the band, and it is more noticeable in the ones that were at warm temperatures. So this gave us information as to that the plasmid was not working correctly, or at least the promoter was not the one that was expressing. So as it was said, we continued to change our approach a little bit, and we started using DH5 alpha cells. So in this case, we used DH5 alpha and BL21 without the plasmid as controls and with the plasmid as our actual tests. But it was also very confusing, the fact that we had bands and at all our samples, which was not what we were expecting. And at the same time, we were running survival tests, and in a similar way, we were getting results that were a little bit weird, because we were getting growth at very high temperatures and at very low temperatures. So something was not working at this point. So we went back and we started looking for what could have been happening. And we got to the conclusion that what we were seeing here was not our protein, but was lysozyme, because lysozyme has a very similar weight as to the protein that we were looking for. So we ran another gel using lysozyme as a control and in the same conditions that we already did. And we found that the bands were very similar in all of that, although it cannot be very clearly seen, but they are there. And in general, we got to the conclusion that we were seeing lysozyme all this time.
Okay, so finally, as you can see, we realized that our methods were not working that well. So we decided to change our vector from pickled, that just as a reminder, pickled functions with cold temperatures. So we decided to change it into PET23A+, which this is a that this vector has a constitutive promoter, which is a promoter that it will always express the protein. In this case, we decided to purify the AFP gene from the previous vector and purify the PET23A+. And finally, we decided to do a ligation.
So we can see the results of the subcloning here. First, we wanted to check the integrity of the pickled vector here. And since we saw solid banding here, we concluded that it was fine. So we continued with the subcloning, where we purified TMAFP and PET23A, the vector we were going to place it in. Of note, however, is TMAFP here. We were expecting to see a band around this region. Unfortunately, we didn't see one in the gel. We did check with the nanodrop to make sure that there was DNA there, and it was confirmed that there was DNA there. So we continued with the ligations, and then we plated those ligated cells, and the results of which you can see here ended up with no growth. Now, discussing possible theories on what had happened, we have a few theories. First of which is the reversal of the gene in insertion. It's possible that when we inserted the gene into the vectors, they went in reversed, and therefore the proteins that were supposed to come out didn't come out right. And as you know, as a protein, the function is based on its shape. It doesn't have the same function. Second off, we have low DNA concentrations, as we saw previously in this slide. That might have been a reason why it didn't show up either. Thirdly, we have power blackouts, which affected both ligase and AFP, because ligase and AFP are very sensitive proteins and they need to be kept at a constant cold temperature. Unfortunately, during the semesters we've been working on this, the power has gone out, and it's possible that the temperature inside the fridge has risen past its acceptable point and resulted in some damage to either ligase or AFP. It's worth noting that AFP is usually worked with in a cold room. However, we don't have that yet. We hope to have some in the future, and that might fix these two problems. Next up, we have lysozyme, which, as we said previously in the other gel, we thought we had the AFP protein, but unfortunately, it just turned out to be lysozyme. Lastly, we have the BL21 versus DH5 alpha question. We originally started using BL21 because it was more apt for protein expression. However, seeing the results that didn't work, we consulted the literature, and it was revealed that DH5 alpha was more successful with scientists who were working with TMAFP.
Okay, so as everything in science, we have a really long way to go. This is obviously not a finished project, and we need to explore more approaches such as doing PCRs, but we are waiting for the primers. And also we are open to suggestions, and thank you so much. Thank you.
Does anyone have questions?
Okay, so first, how interesting, you guys. I always love coming to these things because I learn a ton of things about stuff that I have no idea about, like beetles that don't freeze. So I wonder a little bit about your methods for the initial part of this. You say that you ran a blast, but was that a nucleotide blast or was that a protein blast? What did you use initially?
It was a protein blast.
It was a protein blast.
Yes.
And is there any way that you, what did you use as a cutoff in order to determine which sequences retrieved from that blast you were going to then use to build your tree? Or did you use all the ones that were retrieved from the initial option?
Well, first of all, what we did is to determine the ones that had the higher percentage of identity.
What was the higher?
Like
you use a cutoff
like
point?
100%, because we had like a range between, if I don't remember bad, between 80 and 100. But as this is a really weird protein because there were not that many sequences. So we decided to expand a little bit further, and we decided to choose some from 60 and 70.
Okay, and you chose the ones from 60 and 70. And that's it? There are no other homolog proteins in all of the entire, eukaryote scenario from your blast? I wonder because there are currently, for example, organisms and plants that live in these very, very extreme conditions in the cold. Yes. So don't they have a protein that shares some of these motifs or domains, considering that you did a protein blast, and so you should be able to see that?
So actually, yes, because well, from literature, we know that many animals and plants has the protein, but from the blast, mainly what it it was only beetles and insects. And I think there were like 2 plants.
Also, we had some proteins that were hypothetical, so we don't know for sure if they if they are that sequence. So we limit our search to the ones that we do know that we have. So probably we do have more proteins that have these antifreeze capabilities. However, we are not sure because in the database we did not get like more sequences.
Sorry.
There's a lot of them, and they have the same function by different methods, different structures. So these ones in particular, often molecules are very rare. They're abnormal, but they have other structures, and we cannot compare them
Okay. Okay, how interesting. I was gonna ask something else and now I forgot. Yeah, go ahead with Zoom because I lost my train of thought.
Yeah, I just want to add what Professor said. We explore some other antifreezing proteins. Some of them use, some of them use minerals. Some of them use other kind of domains because we explore on a domain called DUF, that it's used that it's present in many antifreezing proteins, but mainly in fish, but not beetles. And this, as Professor said mentioned, this antifreezing protein is like very particular for insects. I mean, we from our blast, that's what we found.
That's exactly where my question is going, because if there are other types of antifreeze proteins that are used in other types of organisms, why did you narrow it down to these ones that are very limited from this very particular, type of beetle that has very few sort of associated sequences for you to work with?
Yeah, actually, that's because it's easier because what we explore is the history. So for the reconstruction and the modeling of the ancestors, because what we did is first obtain the similar proteins from other, and we found that it were found in other insects. And that we make all the methodology to obtain a tree, phylogenetic tree, and from there we obtain the ancestors. So it's easier for us to work with a smallest model, and also for the model, it's easier to work because if we explore in many different species that are maybe apart for millions of years, we need a lot more of samples, and that will not just only take time, but also because you have to put this in the page, for example, and this information needs to be processed, and it could take weeks, months, depending on the amount of information, and also because there are in the Quake, there are many other works. So for this research, we focus in this specifically on only in insects because it was easier and faster, and the evolution history, so it's shorter.
Okay, so you felt it was better to work with a few amount of samples in order to build a story more time efficiently. But I remember my question. I'm sorry, I really do wonder about this. You say, or I'm not sure I understood it correctly, but in one of the slides, it says that one of the future perspectives is to improve or enhance the sequence using the ancestral. Why would you think that using the ancestral, you could enhance what has, come out of your analysis?
Yeah. So from the ancestors, as I mentioned, we found that they were more stable because AFP, as also in the last presentation we mentioned, it's very unstable under normal conditions. So we found that. So maybe checking which changes we can make for the engineering, achieve a better protein to use in the industry. For example, if you want to preserve organs, you need to it is under some conditions, and if the protein denaturate, it will not work. So we need to make it more resistant at first. And you asked me something else already about yeah. And also, for example, from the ancestors, we can learn that the isoelectric point, that's something even more interesting, and even the hydrophobicity. Because if we want to apply it into tissue, what kills you is not the freeze by itself, it's the crystals that form inside, right? So the crystals penetrate the membrane and destroy the cell. So having one that can achieve to pass the membrane and also remain in the same conditions in the pH isoelectric point will be easier. So that's why we are basing on the ancestors, because they are better in these points.
Not necessarily for everything else.
Everything else, industry, for preservation, food preservation, and I don't know if anyone wants to add a little more.
The only antifreeze protein that we know that is hydrophobic. And if it's hydrophobic, that's no reason. It can be easier for you to get inside. So that's the real reason. Not actually because the low number, because there are many hydrophobic proteins that we know of, therefore that's very helpful.
Thank you so much.
I think it's an amazing work. Maybe we can continue later on. I'm sorry that I'm cutting you down, and it's, otherwise we're going out of time. Sorry.
Okay, so for the next presentation, we'd like to invite Daniela Tapia Naranjo to the stage to talk about her presentation on novel product, versatile hair mask for cell.
So, hi everyone. My name is Daniela Tapia Naranjo, and I'm going to present versatile hair mask for sale. So first of all, this is my table of contents where I'm going to show my introduction, methodology, results, discussion, conclusion, my acknowledgements, and references. So Vercel is a hair mask that I made with natural products like rosemary and orange leaf oils that was extracted with a steam distillation. And it functions generally as a moisturizing cream, also a hair mask or a conditioner for all hair types. So we know that in the market, there are a lot of products that had very toxic chemicals that can cause cancer. For there is some example of them as parabens that and sulfates that are the most common in the products. And parabens are most known because they mimic an hormone that is estrogen that makes the hair grow. So this can cause a disruption in the hormonal balance and can cause hair loss. There's also the phthalates, the formaldehyde, that it is very often used in straining hair treatments, and this with further usage can cause cancer. There's also dimeticon and polyethylene glycols that has the same effect as parabens, but it is another chemical. And this hair mask, that's why I decided to do it with natural products and safety chemicals. So the most important ingredients that I introduced to the hair mask was the rosemary and orange leaf oil that I extracted with a steam distillation, and these can have very good benefits to the hair as growth hair, even better than minoxidil, that it is a medical there is a medical transcribed by medicals to the hair growth, and also the management of bacterial infections that can cause hair loss, and also moisturizes hair and prevent it to turn it rough and make it look more healthier. Also, there is the coconut and argan oil that protect the scalp of the hair and moisturizes, softens hair, and increases also the shine, and reduces the appearance of split ends, so it makes it also look healthier. In my methodology, here are all the ingredients that are included. Is the coconut oil, the argan oil, rose of water, vitamin E. There's an auto-emulsifier that I introduced that is vegan and has cetearyl olivate and sorbitan oil that becomes from olive oil and sorbitol. These are very good ingredients for moisturizing the hair. And also the natural rosemary and orange leaf oils, conservative coast guard. There is a safe chemical used in cosmetics, and it is used instead of parabens and sulfates that are also considered conservatives. And it is more safe, and it is used in 0.1 and 1.5 concentration, so it is not as hard as other chemicals. Also collagen, shea butter, distilled water, and that's all. So my methodology, I use a video that I found on YouTube, and I use that steps. So I use that video to do my hair mask steps, but I optimize and introduce new techniques to do my hair mask more efficient. So here's my methodology. First, I do an oil extraction that was made by a steam distillation, and I use 150 milliliters of distilled water and rosemary and orange leaf oils. And there was obtained 50 grams of rosemary oil and 30 grams of orange leaf oils. Then I mix both of them together, and I obtain, as it shows in figure 2, the rosemary and orange leaf oil after the distillation. Then there was to be 2 phases to do this cream, so the autoemulsifier can work. So first we have the aqueous phase and the oil phase. And in the aqueous phase, we have the liquid ingredients as the rose water and the distilled water. Then they were put it in double boiler with the oil phase that contain all the oils mixed together and the emulsifier. And we put it double boiler so they can be melted, all the oils, and also the liquid ingredients be hot. Then we make the thickening part that both phases, after they were double boiled, they were left to cool for 5 minutes and then mixed together at 70 degrees Celsius with constant stirring. And then after stirring, they were left to cool. Then I decided to put it in the flask that I was going to put it at the end so it can be thickened. Then after a few hours, the last ingredients that I add were 30 drops of conservative and 25 drops of vitamin E and 50 grams of collagen. That are the additives. And here you can see the final product. Then in my methodology, I also decided to do a sensory evaluation with 20 random panelists. These panelists were not trained because I was doing this sensory evaluation here in school, but I give up to them 10 grams of sample so they can try it in their houses. And then after that, I give them a questionnaire using hedonic scales so they can evaluate different characteristics as tactile texture, appearance, texture of the hair at the end of the day, texture on hair after applying, texture on dry hair, and aroma. Also, I conduct a consistency analysis. This was made in Cicata, Querétaro, and in the laboratory of biochemistry, and I use a rheometer. That it is an equipment that analyzes consistency and viscosity. And the sample was placed here, and then this apparatus goes down, and we apply different parameters so it can make us a graph that analyze the behavior of 2 samples that I was analyzed, one with the original quantities and one with 80% less of the quantities, to see if we can produce the hair mask at a lower, cheaper cost. Then for further investigation, I was thinking to make an analysis of the color, and this analysis is made with a colorimeter apparatus that it is this one. And this stands for, the results give us the luminosity, saturation of the color, and the hue of the samples. And it is expected that the hair mask give us a color between white and pink palette. And with these values, we can see it in the diagram where it extends the color. So in my results, I made a radar plot that evaluate the characteristics that I mentioned before from 1 to 9. And as it was expected, it had very high values. For the aroma was 7.46, tactile texture 6.85, appearance 8.5, texture on the hair 7.96, texture on dry hair 8.32, and at the end of the day 8.32. So this means that all the people give like an overall acceptance to the hair mask. Then in the consistency and viscosity analysis, we have the graph that the rheometer made, and this label stands for the viscosity, and these lines mention the behavior of both samples, that at the end the viscosity goes down. So this means that the behavior that the hair mask has is going to be disappearing in the hair when you apply it. And it also shows that it behaves as other hair masks in the market. For the discussion, I saw that the overall acceptance for the hair mask had like very high values. So we can see that the final product has very well been received by panelists. But we are making improvements with other physical chemical analysis to be more assured to have a more well product. And the consistency and viscosity analysis reveal no significance in both samples, so we can know that we can make the hair mask with more cheaper quantities of the ingredients, so it can be less expensive to produce it. And the rheometer confirmed that, as I mentioned before, it was a non-Newtonian fluid, that it is shown that when you apply it to the hair, it's going to disappear as other hair masks. In conclusion, our product has an overall acceptance to the public, so I hope that in further future it can be introduced to the market. But we are going to do more physical chemical analysis to improve our hair mask. And now I'm going to thank to Arkansas State University for letting me present and letting me make this project. Also to the special topic course and Dr. Manuel Santos Basurto and Dr. Paola Camacho for guiding me in this process. And also particularly Dr. Paola Camacho, because he was mentoring me in this process and let me the opportunity to collaborate with her, and also to Cicata Unit Querétaro for granting me the permit to use their stabilities, their facilities, and lastly to my family and friends that support me in this process. Here are my references. Thank you.
I'd like to open the floor for questions.
So thank you, Danny, for your presentation. I'm quite interested on your results. I saw that you have your flasks over there. If you could share it with us. But my main question, I know you have the essential oils by steam distillation. Do you know how many grams of your raw material do you use in order to achieve 30 grams or 50 grams that you managed to achieve? Because I know you may use a lot of raw material.
Yes, I use a lot of it.
Do you know how many? How much?
I didn't weigh it in particular, but I use a lot of rosemary, and the round bottle flask was around 100 milliliters, and it was like at the half of the raw material, and also orange leaves oils. So it was a lot, and I run the steam distillation for a lot of time. So that's why I also obtain a lot of product.
Okay, thank you. And do you know or have you compared your product to the original one, the one that you were gathering from your video?
Sorry, I didn't hear
Have you
you very well.
compared the original hair mask that you were using the recipe originally against your product? I mean, have you compared against and I don't know, like
With
in
other
all
hair masks?
like in like in chemistry, like in all the trials that you have already run, have you done that?
I was going to do it with the rheometer to compare it with another type of hair mask in the market, but I didn't have like the time to do it a lot of time. But it is like a purpose of me to analyze it with other type of hair mask because I want to see also with the hair mask that promise that are natural products. I want to make also to them a physical chemical analysis to see if they are equal to my hair mask or my hair mask is even better.
Thank you.
Test also the rheometer with the mask of Sedal, that is a commercial mask, but which was the main situation with that mask that failed the test, and with what reason that we could not do it. More than we can have time, I think what behind another reason.
Yes.
And I think he also is asking related to the percentage probably, no? Which is the percentage in terms for, I think you have 100 milligrams, no, 100 grams, sorry, but which is the percentage of oil that 100, not 50 grams percentage, and also the reason behind, like why it's not possible do the characterization with commercial mask.
Yes, it was not possible to do the characterization. I made it with one Sedal hair mask, but it was like very liquid. So when we put it in the rheometer, it shows that the viscosity was very, 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 very low. So the results were very weird, and it looked like there had like no consistency. So it was not going to be like well compared with my hair mask because my hair mask is more thickened. So they said that one it was more liquid. So that's why I could not compare like very well with the my hair mask and the Sedal one. And
We'd like to ask if there are any people online who have any questions.
Okay. Which one is the originality of your hair mask in comparison with the other ones that are considered natural products? I don't want to say the other hair mask, but the one that are coming from natural products. So you can compare there. If it is the methodology in the ingredient.
yes, I think that in the ingredients, because not a lot of hair masks contain rosemary, and I wanted to use this ingredient because it is very well known for hair growth, and many people suffer of this problem. So I wanted to add this essential oil, and there's no hair mask that combine rosemary with orange leaf. And also because it is made of natural products and vegan products, so it is currently free also. And many hair masks also, as I mentioned before, contain many chemicals. So this one assures that doesn't contain a lot of chemicals only because of the conservative. So I think that has natural products and it is not as dangerous as other ones.
And I have a tiny other question, but super fast because I know we are late. How can you be sure that your hair mask it is really doing what you are claiming it is doing? Because you were saying that what was the hair splitting and all these kind of things. Well, the appearance I can understand it, but how would you prove basically all the rest that is not just appearance?
Yes, I made a sensory evaluation, but in for the future, I wanted to make it with trained panelists because these panelists were not trained, but I want to make it with more panelists and trained ones, so they can give me better results if the hair mask is working as I mentioned that is promising, because Yes, I think that we could give the samples as I did before and see with the time that with advanced usage of the hair mask, it could be seen that it really helps to the hair to don't see split ends and be healthy hair. Gracias.
Per- All right, for our next presentation, we're going to be calling up Sebastián Carrión, Diego Muñoz, and Carla Reyes to talk about molecular relations in the signaling pathways of vitiligo and depression as a key for possible treatments.
Well, first of all, good afternoon. And today we are here to present our research project on vitiligo and depression and the molecular mechanisms that they follow in order to develop new biochemical maps. As many of you might know, vitiligo is a disease that is characterized by the appearance of white patches on the skin. Despite not being widespread, it affects between 1 and 2% of the world's population and over 3% of the Mexican population. At the moment, it's not well known what exactly causes it, but it's well known that it could be triggered by stressful situations. Depression, on the other side, is a complex disease that gained visibility after the pandemic. At the moment, this disease is not well known what causes it, but is associated with a low mood. Well, some scientists says that it could be caused by genetic predisposition and also by environmental factors. The environmental factors are the ones that are said to be complex and multifactorial. When we started our research, we began with a review. Then when we doing the review, we noticed that there were not enough biochemical maps of vitiligo and depression, that they were not complete, and there were not even maps comparing correlating these two diseases. Therefore, this generated the necessity to create new and more complete biochemical maps of these two diseases and one that correlates them because there exists a correlation that is it's documented but not explained a lot. Therefore, we began our research on pages as PDB, KEGG, and PubMed. We searched for the molecular mechanisms of this, and then began our correlation of these two by searching for the maps, and then we created our new maps. We selected some we used some filters that the articles needed to be no more than 10 years old, that didn't discuss the immunology of vitiligo, and didn't focus on the statistical analysis, and preferably we selected articles that had maps on it in order to be more easier to compare. Next one.
Okay, so these are our results. These are the maps that this is the first map that we built for this project. This is actually the second or third time that we are showing them, so they are our babies. These are the maps that we built. In this case, this is the map for the myelogenesis process. As Carla says, there are already maps for myelogenesis, however, this is the most complete at the moment. We can start understanding this quite big and complex map with the gene POMC, which creates a precursor protein for certain polypeptides such as alpha-MSH. This alpha-MSH is required to activate the intermembrane protein MC1R. And when we activate this protein, we can initiate the AMPK pathway, which is this one, which is a really important and really complex pathway that is involved in a lot of cellular processes. Now, another really important pathway is the Wnt pathway, which promotes the transcription factors TCF and LEF through the regulation of the enzyme GSK3 beta. When we combined these transcription factors with PrEP, which is produced through adenyl cyclase and the PK-AMPK conjugated pathway, we can activate the MITF promoter. This promoter leads to the production of the protein MITF, which is later phosphorylated by, again, the AMPK pathway. Now you can see why I say that this pathway is so important, and it will continue appearing. This phosphorylation allows us to associate with the MBOX and start the transcription of the enzymes tyrosinase, protein-associated tyrosinase 2, and protein-associated tyrosinase 1. With these enzymes, we can metabolize tyrosine into L-DOPA and begin all the melanin synthesis of eumelanin and phenylalanine, the two types of melanin that we have in our body. Okay. Now, this is the second map that we built. This is the biochemical map for depression. And I need to clarify, making a complete map for depression is extremely difficult due to the complexity of this disease. So this map is based in pathways that have a high relation with melanin and with the melanin synthesis and the tyrosine metabolism pathways. And we can start analyzing and studying this pathway by the activation of AKT, thanks to RAS, another protein of, again, the AMPK pathway. And we can see how AKT actually regulates a big amount of another pathways, one of which actually leads to the tyrosine metabolism through the Wnt pathway that we mentioned in the past map. Another important part, and that is also interesting, is that AKT leads to a promotion of the glutamate synthesis. And as the name implies, the glutamate main function is in the glutamatergic synapses, the main excitatory synapses in the brain. And what we found is that when comparing these maps of the glutamatergic synapses and the dopaminergic, we found a lot of similarities with maps that lead to the melanin synthesis. For example, like the production of PKC through the glycerol and PLC, or showing in this, spoilers, showing in this map the hydration of tyrosine into L-DOPA, which is present in the dopaminergic synapse. Also, I want to highlight all the molecules that are highlighted in green represent those molecules that are present in both of our maps. So we can see this high relation that that melagenesis and depression have. And before ending my part and giving the word to Sebastian, I want us to focus in this, which is the vascular endothelial growth factor, which together with some other factors like the HIF-1A are in charge of regulating angio and neurogenesis. So we can start thinking how disruptions in this pathway can lead actually to malfunctions in the brain development and functions.
Right, thank you. So as you saw, these maps can be really complex. So we decided to make a third map that is not exactly a biochemical map of vitiligo because it will be extremely complex and you would literally see a cobweb in there. So we decided to do this relationship map that showcases important relations between vitiligo and depression. In vitiligo, there are multiple failures in the melanogenesis pathway. The first one being the tyrosine biosynthesis. This happens mainly because of two reasons. So it has been reported that in patients with vitiligo, there are increased levels of homocysteine and low levels of phenylalanine. Homocysteine is an analog of the amino acid cysteine. And what it does is that it inhibits the tyrosine biosynthesis. On the other hand, phenylalanine degrades into tyrosine, and it's key for tyrosine biosynthesis. Now, as it was mentioned in the mass before, tyrosine is really important because it can become L-DOPA, which later on degrades into dopamine or melanin, depending on which pathway it follows. Or it can also go into tryptophan and then into serotonin, which are really, really important for the depression pathogenesis. Now, moving on, we have two master genes that are keys in this process, SOX10 and PAX3. Both of them regulate the MITF pathway. whose importance you've already seen. But they're also really important in embryogenesis. They both direct the migration of cells in the neural crest. So we believe that for our future research, it would be really interesting to look at these genes and see how, if this is the genetic factor behind the predisposition to depression and vitiligo. Finally, PAX3 controls cell function and cell survival. So we believe that it's possible that in vitiligo, melanocytes are really, really sensitive to oxidative stress. And that has been reported in the literature. So we believe that it's possible that this is due to a transcriptomic profile of the gene PAX3 that is altered in some way. Also talking about cell survival, it's really important to highlight the AKT-PKB pathway, which you've seen before. That is also really important in cell survival because it regulates the protein BCL2, which is a protein that has to do with survival signals, and it's an apoptotic regulation. And it does this by activating the transcription factor CREB and inhibiting other pathways. We've also mentioned the WNT pathway, or WNT, as we like to call it, that is really, really important in melanogenesis and in depression. However, WNT also regulates the master genes SOX10 and PAX3. The problem here is that WNT can be inhibited by oxidative stress present in both the pathogenesis of vitiligo and depression. So it's really important to know that oxidative stress can cause a lot of havoc in these important biochemical pathways. Now, knowing all this information, we set off onto looking for treatments and a way to solve it. And doing a literature review, we found 3 compounds that have been tested individually, but never together, and that we think that being tested together would have huge impacts. These compounds are vitamin B12, folic acid, and antioxidant compounds like ginkgo biloba or sulforaphane. Now, vitamin B12 inhibits homocysteine, which will allow us to inhibit the inhibition of tyrosine biosynthesis. Folic acid can be degraded into BH4, which promotes the degradation of phenylalanine into tyrosine. Finally, antioxidant complexes like ginkgo biloba and sulforaphane can help by activating antioxidant regions in the genome, like the ARE complex, by allowing its transcription factor, NRF2, to be separated from its ligand KEAP, who usually keeps this transcription factor in the cytoplasm. But once it's separated from KEAP, it can translocate to the nucleus and activate the ARE complex. Now, I just want to clarify that this treatment has not been tested. However, we believe that it is possible that it can reduce the impact that this disease has and work as a treatment. And while these 3 have been reported individually, they have never been reported together. So we believe it's a really good future research question for this project. Now, throughout all of this that you just seen, we can conclude that the relation between vitiligo and depression goes way beyond just a psychosocial factor and way beyond just how people feel. There is a biochemical connection between these ones.
Well, thanks to the identification of these common factors, we can think about future treatments such as the one that we already present or other ones, and also ask new questions as the relationship of SOX10 and PAX3 in development of the neural crest, and also the impact of each part of the pathways that we presented here in the development of the disease, of the grade of the disease that the people have. Also, it's important to comment that we conclude with this, that if you're a person with vitiligo, you probably will have depression or some mental disorders. Era para. Also, we want to thank our mentor, Master Acosta, for guiding us through this project. We also want to thank Andrea Aguirre for being part of the research group, and a special thanks to Andrea Gutierrez for helping us with the design of the maps. And thank Arkansas State University and CQ-AState for letting us present here. And that will be all. If you have any questions, we will be happy to answer.
Hi guys, I have a couple of questions. First of all, what I find a little bit curious is you mentioned that there were no maps relating both diseases, right? And that's what you decided. But at one point you said that that research indicated that there was a relationship. So how come if scientists from researchers, if there was a relationship, if there were no maps that correlated the disease. So how did they?
Yeah, sure. Thanks for your
Answer
question.
into it?
Yes, well, the thing it has been mentioned, for example, there have been, as you remember our filters, there has been a statistical analysis showing that there is a correlation, like there is really widespread between people with vitiligo depression. And for example, there has been analysis in based in certain of the molecules, like for example, okay, we think that they may be a certain relationship because there are failures or because of AKT is mostly because they not because they have identified the pathways in certain way, mostly because they have seen the prevalence of depression in patients with vitiligo, or they have seen that they, for example, this that we have seen, like teacher these disruptions, or it's like the teacher these disruptions in L-DOPA or in the glutamatergic synapses or in the production of tyrosine, or that they have lower, both of them have lower levels of tyrosine or of phenylalanine. So it's more like for that part, it has been think but not confirmed.
Okay, so it was speculated.
Yes.
Okay.
Yes, but that's the importance, and that's why we make these maps comparing. How they share all these pathways, all these molecules, and how failing one part, these pathways can lead to failures in others, and that way pathways are shared.
Yeah, and I just want to add that there's not one that compares specifically these two diseases. It compares skin diseases with mental disorders as a general, and that's where the speculation comes. And there are, like in the articles that we've read, they have sections for this skin disease. Like they compare various skin diseases like psoriasis, like dermatitis. I forgot the name, but different skin diseases. And they just look at different levels, as Diego said, of, for example, phenylalanine or these compounds, but there is nothing similar to these maps that can actually compare them more than speculation.
Okay.
It has always been that this relationship is more likely because of psychosocial factors. You have vitiligo, so you have social anxiety or your. But it's not like something that it's only psychosocial or psychological. It's something that is actually involved in the biochemical and signaling and cell signaling processes.
Okay, that's a little bit. As you have mentioned, disorders are very complex.
Yes.
So in the case of vitiligo, could be also backward that the condition itself is the one that 4-12 depression because of social anxiety and stuff in the but as you have mentioned, these maps, these metabolic maps are really big. You know, they're really complex. These are basically just summaries. Right? They're really complex. So there's a chance that because your regulation is so complicated that something overlap, but actually there's no direct relation to them. So how do you know if your speculations are not being forced to fit your question and you're just seeing the facts for what they are? I'm not explaining myself. Like if it's not wishful thinking, that you're just putting yourself that it's not there because it's a bit vague and a lot of things can overlap. You know, if I pick up, I don't know, any other disease that has nothing to do with this, there's a chance that it's going to be involved at one point. So how do you distinguish between fact and wishful thinking and speculation?
Yeah, totally. And like biochemical pathways are extremely complex. I think we've all gone into the cake side at some point and just see the huge biochemical pathway that can appear. And something that we did not touch because we didn't wanted to look at the immunological aspect of vitiligo, but there are a lot, and by a lot, like there's really way too much of this to just be overlapping rather than coincidental. For example, don't do research on the immunological aspect, but one important transcription factor in immunology is NF-kappa beta, which is super important for T cells. And as you see, this is important also in the biochemical pathway. And we can also, the most studied aspect of vitiligo is immunology. So there are lots of overlaps to just be coincidental. Actually, in this map, everything that is in green is what overlaps with this other map right here. So yeah, and master genes regulate 1,000 things. That's for sure. They regulate absolutely everything. But the thing is that also like these pathways overlap, sections in biosynthesis also overlap. And I mean, you have some of the most important neurotransmitter also overlapping and being important in the melanogenesis process. So it is, well, I understand your question and it's totally valid because we also thought about that at the beginning. We were like, well, I mean, a lot of things overlap in biochemical pathways. So yeah.
I would just recommend that, the approach, I think the idea is good, but in order to avoid saying there's a lot of overlap, because that tells you basically nothing, use statistics. And then you can say which things overlap and are significant and which are not. And then you don't end up speculating. That would be like a step. I know that this is like the first thing you search. You have the idea and it looks good. Why don't you go further and use statistics and just stop saying, it looks like a lot, it looks like this is a threshold and it's significant or not.
Also, while we were researching, we found an article that were talking about the correlation of these two and other mental disorders, but instead they didn't focus on the explanation of the pathways. They focused on identifying a real correlation. They were some biofactors in the urine of the people. Well, they identified in the urine of the people that were increasing as they have a vitiligo in a more developed way, like vitiligo type 2 or so on. And this biofactor was correlated with mental disorders, not only depression, was more related with anxiety, right? So there is a real correlation, not just speculation. There is documented.
The next question will come from online. If you'd like to go ahead.
Thank you.
Marco,
Thank
if you'd like
you
to-
for giving me the opportunity for ask. I want some question for the three presenters. Did you search some information about transgenic mice model of vitiligo, and where this signaling information of these biochemical maps that you are presenting are inhibited, and how will we change the maps in your proposal?
Could you repeat that last part? We didn't quite hear it.
If you search some information about transgenic mice, about vitiligo, and how will we change this information in your biochemical maps, correlation vitiligo with depression? Because there is a lot of information about these transgenic mice in vitiligo.
Yes. No, yeah. So that we have noticed that there is a model of vitiligo mice. I don't quite remember right now, but we did research about it, and the model is made differently. Like they do not alter biochemical pathways; they remove certain genes. However, it would be really interesting to use also those models to do transcriptomic analysis, both of the genes that were mentioned, and also to measure the different things that we can see here in the map, to also perform statistical analysis to see the real transcriptomic profile and how affected this is in a statistical manner. I don't know if that answers your question.
Okay, thank you. Yes.
Actually, part of our objectives to have future with this project is actually trying to experiment with mice, with vitiligo model of mice, and try to disrupt certain parts of the pathway. For example, notice that if they have, for example, all the AKT pathway disrupts certain parts, like for example, the promotion of DSG or the regulation of GSK3 beta, or certain parts of MAP or of the, and okay, or maybe in the expression of these genes for these enzymes, or how this can start leading, or how much is the effect it has in the disease. Maybe, as Dr. said, if we affect certain part, for example, of the MPK, maybe it doesn't have an effect as big as if we affect the inhibition of GSK3 beta or of the activity of the gene POMC. So it's part of our future objectives.
Do you have any other questions?
So for our final presentation, we'd like to invite Jimena Gijon Garcia, Jesus Alejandro Perez Andrade to the stage to talk about the Axolotarium.
Good afternoon. My name is Alejandro, and I'm the co-founder of Axolotarium.
My name is Jimena, and I'm the co-founder of Axolotarium as well. Today, we're going to present the results of our project, Axolotarium. So let's start by giving a brief introduction into the importance of the axolotls. We have 4 main points for this. First is to address their regenerative capacity. Axolotls are able to regenerate part of their gills, tail, heart, brain. Second, they're a model organism due to their anatomy. Axolotls, even though they are in the adult stage, they are able to retain some juvenile traits. As a third point, we have the opportunity, thanks to axolotls, to explore the environmental changes effects in endemic species. And as a fourth point, we have the potential for biomedical research on human pathologies. The objective of the Axolotarium is to implement protocols that maintain a healthy population of Ambystoma mexicanum by providing a controlled environment and ensuring their welfare and conservation. Oh, this is not the...
Before getting deep into our methodology and our results, we want to share you some important considerations that we have to follow. The first one, these are the water parameters that we implemented in our project. Also, in terms of diet, we're going to provide a diet rich in protein, since they are carnivore animals. Also, if we provide living organisms as food, we're going to treat them before to eliminate the possibility of having a parasitic infection. And also, we're more or less giving them the 4% of their body mass every day as food. Also, our initial population was of 8 specimens. They were acquired in different times. And, well, getting a little bit deeper into our methodology, we used this habitat infrastructure. We included several elements to take care of the axolotls, of their necessities and the space they require. And we perform monthly 4 different monitoring processes. The first one are the water test to ensure the quality of the water. The second is material sanitization to make sure that all the tools are clean, and in case of a pathogen in our colony, we don't make cross-contamination. Also, we perform coproanalysis to make sure that there are not parasitic infections in our specimens. And finally, we do a diet monitoring that helps us a lot to know if the axolotls are healthy or not, and to know if they are growing or not. For reproducing a couple of specimens, the first step is just to observe them, to see their physiological and behavioral characteristics. If they are prepared to mating, then we prepare the habitat. The habitat is basically a water tank with hiding spots. We remove all the filtration elements, we remove all the decorations, and we leave them there for some times until the mating ritual is finished. After that, the female is going to start laying the first couple of eggs. So we are going to relocate the male in another water tank, and we're going to leave the female for 3 to 4 days so that she can lay all the eggs. After that, we are going to relocate the female, and we are going to start the recovery process, since the pregnancy cycle It's something that takes a lot of energy from the female. And finally, we reorganize the eggs into groups of 50 in plastic containers. After that is just monitoring the eggs, monitoring the temperature and the water levels of the plastic container. We just have to wait until the embryos develop fully and they start to hatch. Once they start to hatch, we're going to start counting them and see, take care of the statistics of the reproduction, and also wait until the majority of them are born. When the majority of them are born, we wait for 5 days since they still are absorbing the yolk sac, and we perform the first feeding with Daphnia or Artemia. After that, they are going to reach a size of 2 to 3 centimeters, and we're going to make a diet change to living tubifex. Finally, the criteria that we are going to use to evaluate the process of our project are these 5 points. The first one, the survival rate of the offspring. The second one, the survival of the youth or adult specimens. The treatment efficacy in case of having any pathological condition, and the reproduction rate, and the project auto-sustainability.
So now moving on to the results section. We have this section called Develop Protocols. So our team has developed some improvements made to protocols found in literature, which there are not too many because the actual tools are not very well known, or at least there are many modifications made by other institutions. But what we have done is to develop improvements in the protocols, and you can see all of them in a more detailed way in this management plan that is delivered to Semarnat in order to become a PIMS. It's approximately 180 pages made by us. Now we go deeper into weight dynamics, but focusing first on the females. We were first analyzing 2 variables separately at first. We analyzed weight and time, and we found something interesting here, and is the fact that whenever axolotls experience a weight loss, it matched the time where they were facing a disease or they were entering a pregnancy stage. For example, here you can see in 2 axolotls, they faced a weight loss due to their pregnancy, but there's also peaks where can be due to fungal activity, or sometimes even axolotls ask us in a very direct way to change their food because they prefer to keep this variability rather than keep eating the same food for a long time. In the case of the males, we have 3 different conditions based on referring to bacteria or nematodes, which is really common in amphibians. In the case of juveniles, as you can see, they don't start from the beginning because they were acquired much later than the adults that we initially had. But something that we can see here is a drastic peak of weight loss caused by bacteria, but we were able to diagnose this condition thanks to the monthly monitoring of the axolotls. And you can see that this axolotl was able to recover with the pass of the time. So what actually do these graphs tell us? Is that thanks to this correlationship between the weight and the time, we're able to diagnose in a very early stage any condition that an axolotl might be presenting. There's a clinical case, Blue is a really special one. He is a Sylvester male, but he's exposing what is called a bilateral growth. We cannot say that it's a tumor because it does not affect, it doesn't seem to affect his growth, eating or breathing, or even swimming. So we hypothesize that it is due to something viral, because even when we poke those growths, there is no release of any type of liquid coming out of it. So this still remains as a case that needs to be studied further.
Finally, speaking of the 3 successful reproduction cycles that we had, the first one was the father was Crepita and the mother was Manotas. She's laying the eggs. We have to take into consideration 4 different important numbers. The first one, how many eggs did the female lay? Of that amount of eggs, not all the eggs are going to be fertilized. So just a percentage is going to develop an embryo, and a percentage of those embryos are actually going to hatch. And the final number is how many axolotls we still have from that reproduction. As you can see, in our first reproduction, we have a survival rate of 26%. This is quite lower than expected. A normal survival rate is of 50%. But this was due to different things. The first one, we had a bacterial outbreak. We had a certain epidemic in the containers, and it was really difficult to diagnose and treat these bacteria, and therefore it caused a lot of problems there. Also, during the winter, we experienced a couple of days that they were really hot, and that could affect directly the immune system of our axolotls. And also, we have the sudden death syndrome. That is basically that the axolotls born with some defects on their organs, and this condition doesn't show up until they grow a little bit and they die. And we have also genetic abnormalities that is normal in these kind of organisms because they reproduce in a really fast way and big quantities. So the probability of having a genetic abnormality it's bigger, and therefore it can have some troubles. And also we have the mother's age, since the mother that had these babies, well, she was 1 year old and it was her first reproduction cycle. For the second reproduction, the father was the same one, and the mother was a melanoid axolotl. Here we can see that our survival rate increased. That was really good. And also the birth rate was quite bigger. This can be due to two reasons. The first one, the fertility of the female, and the second one, the age of the female, since this was a more experimented female. And lastly, we had a third reproduction that it was exactly the same mother and exactly the same father. This was a really curious case, since normally these animals just reproduce once per year. But due to the excellent conditions that we had in our laboratory, the axolotls felt the freedom to reproduce a second time in a couple of months. And we got this survival rate. It was an 80% survival rate, which is quite good, and we want to improve it in future reproductions. In terms of growth rate, we start to analyze something that was really weird of our reproduction, and it was that by comparing the expected growth of an axolotl, that it's the blue line, we found out that our axolotls were quite small in comparison with the expected growth. And at first, our theory was that we had a mistake in our protocols of feeding or maintenance. So we send a sample to an expert that actually reproduces a lot of axolotls. He treated those axolotls the same way he treats other reproductions. And by comparing the reproductions that he had in his own specimens and our specimens, we found exactly the same decrease in size. This means that it was not a mistake in our protocols, but it's something genetic. So the most probable scenario is that our axolotls have in their genetic lineage a very short variability, and this is causing errors that affects directly the survival rate and the size of our specimens. Here we can see the same in the third reproduction. And here actually we can see that the difference is bigger because probably there, as it was the second reproduction of the female, probably there was some bigger mistakes there.
So for our conclusions for the axolotarium is we state that we have developed a stable colony for the Ambystoma mexicanum. This is thanks to the disease treatment protocols that have been improved and have resulted in the 3 successful breeding or reproductive cycles that we have had. Definitely, endogamy is a significant condition that we need to address in the axolotarium because this comes from something that was out of our control, but it requires further research, which we can do. Water hardness should be analyzed. Why? Because apparently when we were making water tests, it appears that water from university lacks hardness, which is something that axolotls need. So this might be a factor affecting the growth of axolotls. Nutrient absorption in axolotls larvae should be analyzed. This is because there is not too much information or statistics about this, so we can definitely do more research about this. So what's next? We have a collaboration with Mabe to improve refrigeration because we need to keep axolotls under 18 degrees Celsius, which also affects their metabolism and growth. We will add new species. Mexico has 26 more species, so we can definitely add more to the gallery. We can do research on possible fertilizer. Why? Because water coming from axolotls disposal has high levels of nitrogen, which result very useful for crop growth. So we want to do research on that. Research on endogamy, as we have mentioned. Mathematical model for estimating a specimen's growth. The graphs that you previously saw are just the beginning of something that we're planning to do next semester as research using a mathematical model. And the improvement of axolotl infrastructure, we're planning to expand so that axolotls have a better space to be conserved. Improvement in the live food colonies. We want to make this project a self-sustainable project. So that's why we need to raise our own colonies and also to keep the quality of this food because we cannot find them really easily. So here are some acknowledgements that we would like to share with you. Thanks to our team as well that have always been supporting us. And our references. And thank you. Any questions? Comments? Yeah.
Well, thank you very much for your presentation. It's quite interesting. What are you doing with this specimen, the Mexican specimen? For my perspective, do you know which are the current permits that you need in order to have the axolotl here in campus? I mean, Semarnat, our current authority, Semarnat, it's quite I don't know, strict in that sense. And do you know which are the current permits that you need to have?
Yes, so for for actually becoming a PIMS, which means Predio o Instalación para el Manejo de Vida Silvestre, we already have our management plan, and this will be submitted to Semarnat. But if you want to do research or you want to sell some species, you have to process another permit that allows you to do so. But it's way easier than this one. This one took us like 1 year and a half, and it's approximately 180 pages. And it has to be pretty specific about the protocols that you need to follow in the axolotl regarding how you feed them, how you reproduce them, the conditions of your lab. And while the permit for doing research and selling them is not so specific, this one, because they already take into account that you have already submitted a management plan. And that's all the requirements that we need for axolotl.
And do you know if you need to renew it? How long do you need to renew it? Like,
Yes, we need to renew it every year.
okay, thank you.
I believe there was a question online.
Question. I went last week and it was amazing. I spent, I think, 15 minutes there watching it. Really nice. I wanted to ask you one thing. In comparison with other conservation centers of axolots or axolots, which one is the unique part of your methodology, and which one is the innovative part of your methodology? And so just to understand a little bit about that.
Of course, obviously we have made a lot of research for making this project possible. There are a lot of centers that they have a lot of talent. They have people that it's really passionate about this topic. But what we have noticed is that normally people that have axolotariums, they are biologists, they are biotech. And yes, they are really good in research, they are really good in keeping them alive, they are really good in reproducing them. But the issue is that that's not everything in a project. Science is really important, but it's not everything, because, for example, funding is something vital for growing a project like this. And we as biologists, we know that we don't have maybe the skills necessary for performing something big enough. So that's why we found that opportunity here in campus, that we have a lot of talent. We have talent in different areas. So we start to call people to action. And we have a team that it's multi-skills. So that has given the project a lot of different interesting insights that is going to allow us to make better research and to be an auto-sustainable project. So we don't depend just in our own money or something like that. And we don't seek for gaining money with this. It's just research and giving some environmental education.
And talking more specifically about the methodology, we have talked to several experts, and what they usually do for inducing reproduction as a first stage is to do it through hormones. Why? Because they care about reproducing them pretty fast instead of following their instead of reaching the optimal conditions. So the difference is that from this stage is that we do it in a natural way. We first reach the optimal conditions so that our axolotls feel safe and healthy enough to reproduce by themselves. Also, something that we have seen is that the amount of space that other laboratories have for the axolotls, it's not that they're bad, but they usually don't pay a lot of attention to induce hunting, teach them how to not choke with tubifex. So we have like this little program, let's say, of how to train axolotls to hunt. So because we have seen that sometimes axolotls feel depressed whenever they don't have any live food. If you keep feeding them with dry food, they will eventually seek only for the hand that that gives them food. So that's something that we try to induce in our axolotls, to never lose that sense of we're in an environment, natural environment. So that's something that we have improved in our methodology. And another thing is that because live tubifex is really hard to get, most axolotariums outside don't usually give it in a very long period of time. But we try to do so. Why? Because it is very nutritious for our axolotls, and it improves their growth. So that's something that we have improved as well. And for the babies, we have taught them how to look for the tubifex instead of giving them in with the clamps. And they have learned how to detect it because they're almost blind. We need to teach them how to identify their food or differentiate between a predator.
Thank you.
Another question?
Do you have plans to do parallel? I'm sorry. Do you have plans to do parallel research in terms of the species that you're growing as food? Because perhaps you could also do parallel research in that sense on how to optimize this reproduction of whatever species you use as food, or increase their quality nutritionally speaking, right? It's also like another axis that you may be exploring.
Of course, yes. For example, we feed them with guppies. This is a model organism since it's reproduced a lot. So if someone sometime it's interested in making some research on it, it would be really interesting. Also, we have a collaboration with a person that has a colony of meztlapiques. It's like a kind of charal that it only lives in the system where the axolotls used to live. So it would be basically, if we elaborate the collaboration, we can study that species that it's 100% Mexican and it's also endangered. Another question?
Thank you all to our wonderful presenters, and thank you all for coming and watching. We'd like to give a special thanks to Julio Cesar, our representative from Copper here today. If you wouldn't mind coming up. We'll make you present you with the certificate.
Thank you so much.
very much for coming. And we hope to see you all tomorrow at 2. Thank you.